Okay, I think we'll get started. I'm going to apologize. First, I'll say welcome to the Centers of Excellence in Hematology Microscopy Monday. I'm going to apologize because I've lost my voice. Today, our speaker is Ken Dunn, and I'm going to let him take over and just give his talk. You are welcome to interrupt and ask questions or populate questions in the chat. And thanks for joining us this morning.
Okay, so thank you, Diane. There are few enough of us here that I can invite you to interrupt and ask questions if you've got questions at any point in the talk. First of all, can you see? Hang on a second. I got to do a couple of things here. Oh, God. So I'm starting. All right. So what is it? Are you seeing my presentation or my presenter mode?
Presenter mode.
Okay. Let me try this again. Share screen, screen 2. How about now?
You're good.
Okay, great, good. Thanks, everybody. Right, so anyway, like I said, if you've got any questions, if you've got any arguments, anything you want to talk about here, please go ahead. And the talk is not that long. There aren't that many of us. And so I would invite you to drop in and ask whatever questions you have. The other thing, by way of apologizing, I don't actually do light sheet microscopy. I'm very excited about light sheet microscopy. I've learned a lot about it, but I'm in no way an expert. And I've got almost no user experience. So with those caveats in mind, you can either stay for the talk or say hell with this and be gone. So anyway, let's get started. And again, interrupt me. I appreciate knowing I'm not talking by myself. Be really terrific if the thing would advance now. All right, let's see. You see the next slide?
Yep.
Good, great. All right. So it's like a lot of things. This is not a new idea, light sheet microscopy. In fact, it was something I got this guy, Richard Zsigmondy, the Nobel Prize in 1925, where you project light from the side of the viewpoint, and the scattered light was detectable. And that allowed him to actually see object motions and colloids. And this was the first time anybody actually detected sub-resolution particles on the basis of diffraction. Ultramicroscopy experienced a renaissance. I think in the mid-90s, it was kind of like rediscovered, and now it's commercialized. And that was the original ultramicroscope, and this is the ultramicroscope 2, which is available from Levision Biotech. So what is light sheet microscopy or ultramicroscopy or selective plane illumination microscopy? It's probably got other names too. Unlike other forms of microscopy where you project the light either from behind the sample or you epi-illuminate through the microscope objective, a light sheet microscopy involves projecting a light sheet through the sample, orthogonal to the image plane. And if you make that thin, it ends up giving you an image just of that one focal plane, if you will. Now, when you think about, most people think about confocal microscopy when they think about optical sectioning. Of all times to lose my voice. So confocal microscopy is a kind of a really elaborate Rube Goldberg way of getting optical sectioning. The deal is you focus a laser beam down to a point, and that point is conjugate with a pinhole. And so any fluorescence that's excited from above or below, most of the fluorescence doesn't make it through the pinhole. So you end up getting optical sectioning out of that. Now, the deal is, you'll notice you're only imaging a point. So to make an image, you're gonna have to take that laser beam and scan it over the sample and build up an image point by point. You collect it onto like a photomultiplier tube or something like that. So you build a 2D image up by mapping detected signal to an illuminated point. With light sheet microscopy, you project a light sheet all the way through the sample and you collect a 2D image on an imaging detector, like a CCD or a CMOS detector. So they're fundamentally different in how they operate. And this has a lot of ramifications. So since you're scanning the image and you need a certain amount of time at each voxel to stimulate fluorescence, it takes a long time to collect an image. A long time in biology terms, it takes anywhere from, people can collect images at like 30 frames per second, but that's very rare. In generally, you're going to be collecting images no more than 1 to 5 frames per second. That's too slow for a lot of dynamic cell studies. And if you were collecting large volumes, and that's a lot of what I'll be talking about today, it can take literally days to collect large volumes of things like images of the entire brain or something like that. The other thing about it is that in order to go fast enough, you're going to have to integrate for incredibly brief periods of time. Each voxel sees no more than 4 microseconds of illumination. And so that means you're not getting very many photons out. In fact, it's been estimated that most images have no more than like maybe 20 photons. And so in the context of 12-bit digitization and stuff, it's kind of goofy because you've got 20 photons to start with. It also means you've got very limited dynamic range. The signal noise is very low. And the other thing is that in order to get any kind of signal out in 4 microseconds, you got to put boatloads of light in. So you focus a laser beam into it. That induces things like photosaturation, and it's also phototoxic to living samples, and you bleach your sample. And the way that it works is you're illuminating an entire cone of fluorescence above and below the focal point. And most of that fluorescence you discard, you just throw it away, which means you've got unnecessary damage, phototoxicity, and photobleaching, because most of the stuff, you're stimulating fluorescence, and every fluorophore only has a certain number of lives in it. And so by throwing those things away, it's a very wasteful procedure. Light sheet microscopy, you can collect images at 100 frames per second. I mean, that varies depending on the signal, but the point is you're collecting images onto a scientific CMOS. You can easily collect images 100 frames per second. That means you can now capture live cells in multiple volumes per second. You collect a 3D volume, and I'll show you some examples of that fast enough to see things move in 3D. And it also means that if you're collecting large volumes, things that take hours to days to weeks, with a confocal microscope, they can be collected much, much more rapidly. And I think this is a really important thing that this is my emphasis on it, is that there's a lot of enthusiasm about large-scale microscopy. And the problem with it is that a lot of times when people show this stuff, they show one image or a couple of images. And at the end of the day, it's just not practical for most imaging systems to collect lots of, 3 duplicates of 4 different conditions and show them. More often than not, they're just really cool looking one-off images. Light sheet microscopy would be the way that you could actually make large scale microscopy practical. The other thing is that you integrate the signal for a lot longer because you're not having to raster scan. If you're collecting in 100 frames per second, that means you're integrating the signal in each voxel for 10 milliseconds. That's 25,000 times longer, which means you have an enormous dynamic range, much better signal and noise, everything is better. This is not something that's generally appreciated in light sheet microscopy. Is the quality of each pixel's data is much higher. It also means that you don't have to illuminate at such high levels. You can have much lower levels of illumination, trading off that speed, if you will, and is ideal for living samples. And since you're eliminating only the image plane, that means you're collecting all the signal. Again, you're not inducing a lot of out of focus bleaching. You're not wasting a lot of fluorescence. So these are some data from various labs. I mean, I literally show one figure of something that was collected in my lab, not even in my lab, through my lab. Anyway, this is some work that came from Jan Husken's lab. I'm going to mispronounce that name. I hope that. Well, anyway, what you can see is on the top of this is photo bleaching. This is signal level over time over 900 seconds of imaging with either a light sheet microscope or a confocal microscope. And you can see that there's just no damage. And the quality of the image is equivalent all the way from the start to the end. What this has been used for is long-term studies of things like Drosophila development. So this is an example. I believe this is actually from a 3D volume. This is from 3 to 7 hours looking at, you'd like to think I'd know what this is. I only have this as Drosophila development. They went through, and over that period of time, they mapped the fate of each one of these individual cells over time. It's the dorsal view and the ventral view. And so for long-term imaging at 3D, this is an ideal approach. And there's negligible photobleaching over that period of time. There's negligible phototoxicity because development proceeds as it would otherwise. So it's a very, very powerful technique for looking at Drosophila development. It's been used a lot for that. but it's also used in transparent systems as well, like zebrafish. So this is an example of 17 hours imaging of a zebrafish lateral line development. And then the top is 12 hours of microvascular sprouting. And the other thing, as you can see, is actually that the frame rate is high enough here that you can actually see individual cells moving. And I've been tricked by that a lot, but I'm reasonably sure that you can actually see that. And then the speed of this thing is fast enough that you can collect 3D volumes fast enough to do things like capture the beating zebrafish heart. Now, one thing I'm not going to talk about today, and it's a way too big a topic, is how you deal with this data. This is phenomenal. Just the volume is enormous. Rebuilding these 3D objects, characterizing all the information here is a whole nother topic for a whole nother talk that I'm not prepared to give. Another more recent development is something called lattice light sheet microscopy, and this is a little complicated. And I actually hope York would be here so he could correct me when I screw it up. So, but that is being what it is may. This is what the cross section of a Gaussian beam, what comes out of a laser beam, if you will. And this is what happens at laser beam over time. As you can see, it ends up getting narrower and broader, but the whole thing is pretty thick. If you generate, I think you put an aperture in the back aperture, you can make something called a Bessel beam. You're getting this diffraction. Okay, good. I'm sorry, I thought I saw a note. You end up getting these concentric rings. And if you project, this is X and Z, so this is like a cross section going through the screen. And if you project this out into the sample, you can see you get a very, very narrow beam profile, but you get these other rings. And the reason I'm talking about this, by the way, is because the optical sectioning, the Z sectioning, depends on how thick this beam is, right? So this is very thick. This is much thinner, but it's got these rings, these lobes, if you will. If you take that Bessel beam and put it through a spatial modulator, you can eliminate those lobes through diffraction, and you're getting a thin single light sheet. And I want to point out that the lattice that you generate from that, now this is, again, this isn't cross section. So that's Z and this is X. So if you think of this as projecting in, this is actually what you're getting. You're getting this illumination pattern. And if you project that across, you're going to see a solid line. But that lattice, if you will, is very important. And the reason it's important, and this is interesting. If none of you have seen, I recommend watching Eric Betzig's acceptance of the Nobel Prize. Eric Betzig's the guy who invented this. Eric Betzig is also the guy who discovered spatially various forms of super resolution microscopy. But he's always been incredibly dismissive of his work with super resolution microscopy. Very modest, very brilliant guy. But he said, I think the super resolution field is still sorting itself out. But if I have a suspicion that the lattice light sheet microscope, and not Palm, will be the high water mark of my career. He talks about super resolution so dismissively. Like, why would you want to do it? It's dead cells don't matter. So this is a thing he's excited about. And this is probably why. And again, I apologize for the complexity of this. There are two ways of doing lattice light sheet microscopy. In one of them, you actually project this lattice through the sample. So this, every one of those lobes will be coming from the side like this. And you collect 5 different images. There's 7 different images, and you offset them. And I can't explain structured illumination microscopy right now, but basically you use that in a structured illumination microscopy mode to get super resolution. Super resolution, meaning 150 by 230 by 280 nanometer resolution. And you can collect images at 7.5 seconds per volume, per 3D volume. I'm not sure how thick these volumes are. Alternatively, you can take this whole thing and dither it, which means scanning it sideways, back and forth sideways. Very, very rapid. And you make a solid
Diane is trying
beam.
to ask a question.
Oh, okay. Thanks, Ron.
I don't know if you have a cursor that you're using.
Oh, God.
I can't see it.
Yeah, and that's a good question. So how do I get a cursor out of this?
You should be able to just use your pointer.
Well, my mouse is here, but no one's seeing it.
It moved across the screen, so it's
I see it.
Can't see it. Is that better?
there. It is there.
Ah, okay. So it's not showing up in presenter mode.
Thank you.
Great. All right, good. Sorry. All right. Stepping back. Structured illumination mode. You take this pattern right here and you project it through the sample but at 5 different phases. So it's being advanced. You collect 5 images, you get a super resolution image out of it. Alternatively, you take this thing and you just mush it up. You scan it laterally. It's called dithering. I discovered that's what dithering means. You do it so rapidly that you're effectively making a 2D sheet that's very thin, and you collect images much more rapidly, but the resolution is not quite as high. This is wrong right there, because I'm saying it's the same resolution, so ignore that. I'll correct it. But you collect images much more, and in fact, I've got it backwards. So, it's 1.5 seconds per volume, that's correct. But check this out. These are examples of movies that Betzig presented in this paper, and I recommend looking at this paper, looking at actin projections coming out of cells. And on the left, you can see you've collected 300 versus 50 sections, but you get higher resolution on the right, but you get 3D volumes collected incredibly rapidly on the left. And the other thing is you're not seeing any evidence of any kind of damage.
I'm learning about this.
Okay, sorry.
I see these things waving in the breeze, but in the real world, what are we actually seeing here?
This is, and I'm not in a position to say what these are because I'm not an actin biologist, but this is fluorescent actin.
So it's inside the cell.
This is inside a cell. These are living cells waving projections around. And is there anybody on the call that can talk to us about what these waving projections are, then we can get at that. But again, this is not something you would be able to detect in any way. This is 4D data. This is 3D data over time. There's no way you could get this without the kind of speed and resolution you're getting from this technique.
And of course, it's in a living cell too.
Yeah. And in fact, that was one of Betzig's major points, is that as far as he's concerned, if any work with fixed tissue is baloney. Everything changes. You need to be doing live cells. And so, like I said, he was way dismissive of the stuff that he's done with Palm and that. Here's another. This is just mind blowing. And again, I apologize because I don't understand the biology. This is Drosophila dorsal closure, and this is actin again is labeled. But check this out. So again, I want to point out that these are volumes being collected incredibly rapidly enough to characterize motion in 3D. And it's pretty astonishing. And not only is it fast enough to collect this data in 3D, it is fast enough to, you're not actually seeing any photo bleaching damage from it either. I had a third example, but I got rid of it I recommend that you go find this paper, download the movies because they are jaw-dropping, and there are more of them. And in fact, one of the things he goes into that I took out of my talk was the analysis. Following individual objects, and I think it was microtubule endpoint markers or something over time. But again, the whole business of analyzing this much data with this much complexity and motion and is mind-boggling, and I think it's probably the next frontier of this work. So, getting back to more mundane aspects of light sheet microscopy. I mean, I think we all appreciate the fact that tissue scatters light. So if you're putting in this laser beam from the side, there's nothing magical about light sheet microscopy, and it still has to deal with scattering. And so this is an example of some early images that we collected where we were illuminating from one side. You can see you get terrific labeling over here, nothing visible over here at all. And the other thing, and that's because the light's simply not penetrating into the tissue. There you also get these shadowing effects where depending on where an object is, I'm sorry, I'm goddamn mouse, I'm sorry. So this is that sample I was telling you about where you have shading from one side. This is an example where, and I don't know what the objects are, but you can see these lines, and those reflect the fact that the light is being shaded at various by objects in the light path. And so you get these artifactual stripes. So the way you get around that, pretty much every light sheet microscope, and this is another example. This is one of the first papers from 2004 when it was kind of like rediscovered by Ernst Stelzer. What you can see is that this side is very bright, that side is very dim. This is a wide field microscope volume. This is a light sheet microscope volume with illumination from one side. This is a light sheet microscope volume illuminated from two sides. So you can see you can compensate for that effect. So there are various ways of doing that. You can rotate the sample, like what Ernst Stelzer did. Leica came up with a system where you have laser beams that are reflecting off of mirrors and then projected onto a sample that is mounted in the middle there somewhere. And you can collect a 3D volume out of that. You can illuminate from that side and that side at the same time. This is what the illumination looks like from one side. This is what the illumination looks like from the other side. This is what the illumination looks like from both sides. And you can see darkness in the middle. We'll get back to that in a second. And this is the same kind of volume that was collected using the twin flecked system. We worked with this thing a little bit. I have some thoughts about it. We can share probably offline. Most common approach is one. This is easy. The LaVision ultra microscope is easily the most deployed light sheet microscope. And if you've got a light sheet microscope at your university, that's probably what it is. And it involves illumination from 6 different directions, and then fusing the images together to generate one image. Bruker makes a bunch of different light sheet microscope systems. Here's one, I guess it's the LCS system that you get illumination from both sides. You collect an image here. Alternatively, you can take this whole thing and rotate it 45 degrees and illuminate from one side and collect an image here. Also illuminate from one side and collect an image here. So these objectives serve 2 different functions. And that's important because you can improve the resolution. So if you think of, let's say we're imaging from over here and we're projecting a light sheet from down here. Well, we all know that we've got poor Z resolution, right? So this is what the cross-section of a sub-resolution particle looks like in the XY dimension. This is what it looks like in the XZ dimension. But this objective over here, this is the Z dimension, and that's the XY dimension. If you take these 2 images together and fuse them together, you end up getting a higher resolution. In fact, you can get isotropic resolution, which is a very cool thing that a lot of microscopists are looking for. And this is something that has been deployed in commercial systems, if you're interested. So here's an example of I labored to find some examples of images that were relevant to you guys. And to be honest, since I'm not a hematopoiesis person, I'm not even sure what matters to you guys, but these are images of the mouse calvarium that were collected. And this is one of the most stunning volumes I think I've ever seen. It was collected with the LaVision ultra microscope system. I'll explain what this means. The point of this volume is to show you 2 things. What happens when you illuminate multiple sides, but also what happens when you clear the tissue. And again, I'll explain that to you in a second. But this is the 3D volume of the mouse calvarium. And we're going to spend some time just watching it because it's gorgeous. What are the labels? White is ostrix, which apparently is skeletal progenitors. Red is endomucin, which is endothelium, and green is also endothelium, which is CD31. And so there are 2 things you get out of this. First of all, the scope of the image. This is the entire calvarium, I believe, and it is imaged at cellular and subcellular resolution. What they're showing here is segmentation of various regions. So you do image analysis to segment out different kinds of objects. And again, this is an enormous amount of information you're going to be dealing with here. Anybody have any questions as we're just gawking at these things?
So
gathering more audience in one of the places.
So Ken, I have a question. This is obviously epitope tagged proteins in the mouse.
I think this is immunofluorescence, actually, but yeah.
It is immunofluorescence.
I believe that it is. I should know that, but I don't. Some of the other ones will be. And I think what you're going with this is how the hell do you do immunofluorescence of big, thick tissues? Yeah, right. Well, we'll talk about that, all right?
Okay.
I'm sorry, I apologize. I think this is actually immunofluorescence. And so we'll get to that issue in a moment.
Great, thank you.
This is another example, a less compelling example, but it's everything I've got. this is autofluorescence and anti-CD31. Okay, so if this is anti-CD31, this is going to be labeled in vivo. In other words, this is an injection. And a lot of times people that do the labeling of vasculature, they do it by in vivo injection while the animal's alive. And so that's one way of getting away from the problem. What Diane's alluding to here is that it's all very fine to have a tissue that is millimeters by millimeters by millimeters and image it, but how the hell do you get antibodies into it? And how do you rinse them out again? And how do you put the next set of antibodies in? You rinse those out. So there are challenges that to how you actually label these tissues. And one of the ways of doing it is actually using the animal's microvasculature to label the endothelia. So pretty much every volume I show you, we think, has endothelia because you're cheating. You're using the perfusion to label it. This is an example of brain, and I don't know if anybody here cares about brain, but it's a gorgeous volume. And I want to point out this was collected in less than an hour. And to collect an image at 4 by 4 by 4 micron resolution of the entire mouse brain, it would have taken 40 hours by confocal microscopy. This is an animal expressing TD tomato 5.1, I think, which is a neural marker. And the other thing that's interesting about this guy, Adam Glazer, who works in John Liu's lab, is incredibly skilled with Imaris and visualizations, these techniques, which are really hugely important for visualizing any of this data. So its point I was making earlier is that it's all very fine to have these gorgeous large 3D volumes that you collected with confocal microscopy. But at the end of the day, if you're doing a research project and you want to compare 3 different conditions, 5 replicates of each 3 condition, you can't have something that's going to take 40 hours per sample. That's just not possible. I mean, it's absurd. So this light sheet microscopy arguably is something that would allow you to actually make this kind of technique practical for comparing things with reasonable numbers of samples. All right. So let's talk about tissue clearing. Tissue clearing, again, is another old idea that's been rejuvenated. At the turn of the century, there's this guy, Werner Spalteholz, who wrote a paper called On Making Transparent Human and Animal Tissue Samples. He put together the samples that he got funded to put these in this show, Human International Hygiene Exposition in Dresden. It took 2 years to produce these samples. This is a cleared human hand. That he actually cleared himself. And he's using some clearing agents that are really similar to what we're doing now. So although lots happened since the early 1900s, and I think everybody's aware of the fact that there must be 50 different ways of clearing tissue, it's not really something that's new. In fact, it was even older than this, because H.G. Wells noted that, in fact, what he said, in fact, the whole fabric of a man, except for the red of his blood and the black pigment of hair, are all made up of transparent colorless tissue. Transparent thing becomes invisible if it is put in any medium of almost the same refractive index. So he knew this even. So this is something that I don't want you to try to commit to memory, but this is the process by which tissue clearing works. So basically, you fix the animal and you do one of several things. You bleach the tissue for autofluorescence, you decolor it, you decalcify, and then you take one of 3 different techniques to process to do clearing. And these different techniques really boil down, all these different techniques really come down to 4 different strategies. One is where you remove the water and increase the refractive index of the medium you're putting in to match protein. Second one is is very similar, but you get less protein loss and you decrease the tissue refractive index. The third is one where you remove lipids and you decrease the protein refractive index. And then the last one is you just incubate them in goo whose refractive index matches the tissue. But at the end of the day, what you're trying to do is you're trying to remove the change in refractive index goes between the mounting medium, if you will, and the tissue. And tissue has a refractive index of like 1.5. Proteins have a refractive index of 1.5. So you put it in something that has the same refractive index, it becomes transparent. You get light penetration. So here's an example of, and I guess I should probably start the movie. It takes forever to play this movie. Here's an example of anti-CD105 and anti-SCA1. So here's an example of a hybrid strategy. They perfused a live animal with anti-CD105 to label the vasculature. Okay, and the animal's alive, and the antibodies go everywhere you've got vasculature. They then perfuse the animal with PBS-EDTA, perfuse with PFA. They post-fix an paraformaldehyde, incubate an EDTA, and incubate with an anti-SCA1 antibody. And this is going to be directly labeled because, as Diane has alluded to, it takes forever to get antibodies to penetrate into a tissue. And in fact, the total time, and then you dehydrate a methanol series and you clear refractive index in BAB, which will match the refractive index of this tissue when you're done, so that the tissue that remains after this process has the same refractive index as the mounting medium. Total time to do this is 6 days. Right, and this was done with a custom light sheet microscope system. Here's another example of this different lab, same kinds of techniques. And this one up here, what are we looking at? SCA1 apparently is stem cell antigen. So it's a marker of primitive hematopoietic cell stem cells. Here's another example of someone who's looking for megakaryocytes. And so anti-GPIX is a megakaryocyte marker. And so you can see the vessels, which are, this is in the segmented volumes. This is after having a computer identify the boundaries of things, which pretties up the images quite a bit. This is an original image, this is the segmented image. CD105 is the vasculature. The gray is bone, which is based on autofluorescence, and the green is the markers, the megakaryocytes. Here's another example. Now, this is collected with 2-photon microscopy, but I apologize for that, but it is what it is. There's another example of doing 3D microscopy of cleared tissues. This is based on ethyl cinnamate, which is very cool because it's not toxic. It's relatively fast, and it ends up giving you something, the refractive index very similar to the protein. So in this case, again, incubated with anti-CD31 antibody, fix the tissue, label with primary antibodies. These guys actually labeled with primaries and then secondary antibodies, dehydrated it, and refractive index matched ethyl cinnamate. Total time for this took 20 days, which is a lot longer. And this one I don't have. This is another example, I think, from the same paper, and so it's probably going to be pretty similar, but it's a cool looking volume. Again, does anybody have any questions about any of this? Well, good. And again, I apologize. I keep on using the cursor on my screen and ignoring all of you. I'll try to stop doing that. Another example of 2-photon microscopy, this anti-SMA smooth muscle actin, which labels the vasculature again. So how did they do it? It's interesting. Oh, it's TD tomato. Okay, so this is genetic model. Okay. And again, you can see all the vasculature in this TD tomato expressing mouse. And SHG is second harmonic generation. And that's actually, that was in some of the other 2-photon volumes as well. It shows you the collagen on the surface of bone. This is another microscope system that actually Jorg, our own Jorg Gersdorf, he's somewhere in here in this list of, no, he's not. How about that? Well, he worked on this microscope system. This is light sheet microscopy of anti-c-KIT, which is apparently a stem cell marker. GFP-Thy1, which labels neurons, and blood vessels, which are labeled in gray. So again, this is a system I'm going to return to because the resolution of this is pretty astonishing. And this is a custom light sheet microscope that came out of Texas, Rado Fialka's lab, who does some really, really interesting work. Oh, and I guess the reason I'm showing this is a third method of clearing called Pegasus. And Pegasus, I guess I didn't show the method. Yeah, here it is. You actually perfuse the animal. Everything is, you're perfusing, you're doing the clearing in the intact animal. So there's transgenic mice, you perfuse with heparin, you perfuse with paraformaldehyde, you perfuse with qualdrol, which I forget what it does. I think it clears bone or something. And then you're actually delipidating, dehydrating, and clearing all in the intact animal through the vasculature. And the thing about this is just the bone marrow, but everything in the body of this animal has been cleared to a more or less extent. And this is an example. Okay, so the other, I'm going to talk about a couple issues. The first issue I talked about was the degradation of the light sheet, the fact that light has to go through the sample, and how do you get around that? And you get around that by either by illuminating from multiple sides, clearing the tissue. But the other thing that people need to appreciate is that the resolution is inversely related to field size. Here's an example of a light sheet microscope volume that was collected by light sheet microscopy, and the same volume collected by confocal microscopy. You see, you don't have very much resolution here as compared here. And this is an example of this is some work that we did in collaboration with the people in Washington, John Liu and Joshua Vaughn's lab. We collected a volume at 5 micron lateral resolution. Let me collect the same volume at 500 nanometer lateral resolution. So these are a bunch of glomeruli. And this is the vasculature labeled with, I think, CD31. And you can see all these glomeruli and blood vessels. And the red is the labeling of this albumin that's been taken up into proximal tubule endosomes. And you can see that you can find the glomeruli, you can quantify the glomeruli, but you can't really see any of the cell biology. That same volume, there we go. This is scrolling through the volume from the top, from the bottom up to the top. And at some point we'll encounter glomerulus. There we go. These are all glomeruli. And the red spots surrounding glomeruli are endosomes of the proximal tubule. So you're detecting individual endosomes inside this gigantic tissue. But again, this resolution that we're getting here comes at the expense of scale, if you will. And the reason for that, the first part of it is kind of obvious. I mean, everybody appreciates that low magnification objectives, unless they're enormous, have a low NA. And if they have a low NA, they have low resolution. So for a 25x objective, you got an NA at 0.95, great. You got lateral resolution of 340 nanometers, and your field of view is only 295 by 205 microns. If you go to 5x, your lateral resolution goes up to 2 microns, but you have a much larger field of view. That's just optics. But the other thing that I think people don't necessarily understand is that there's a trade-off of field size that's dictated by the properties of the light sheet. When you project a light sheet through the sample, if you throw on a low NA objective, you get a very, very broad field of view, but it's very thick. Whereas you have a high NA objective, it focuses down to a very narrow band, but that narrow band is very thin, very narrow. And I'll explain this. It's not very long. And the field of view from a thin light sheet is limited to something called 2 Rayleigh lengths. And so because of that, and this is where I'm going to screw up my explanation, if you have a low power illumination objective and a low NA objective, you're going to have something that's got it's going to be very thick. It's going to be 15 microns thick beam, which means that's your axial resolution, but a very big field. You're going to get 3 millimeters of field of view from that at 15 microns thickness. If, on the other hand, you use a higher NA illumination objective, your resolution is increased by a factor of 3, but your field size is reduced by like a factor of 10. So there's a trade-off between the two of them. You can collect the images very rapidly at low resolution. And so for a 267 and 280 micron field, you get a resolution of 340 by 340 by 4.7 microns. 1200 micron field, 1.1 by 1.9 microns. Now, one way you can work around them, this is a very exciting system. I mentioned earlier that Radar Fialko's lab in Texas, that if you take the light sheet, and rather than just projecting a light sheet through, you actually focus it down and raster scan a beam across. You can focus that beam to different depths into the sample. And so rather than just accepting the thickness of this beam as a given, what you're going to do is you're going to scan that beam. You're going to focus that narrow region to various distances. I hope this is clear. Distances from the illuminating beam. And by doing that, you extend the depth, the width of the region that you can image at high resolution. So you scan an illuminating beam that's focused at different distances, and you collect a fluorescence on a CMOS detector whose rolling shutter is synchronized. So a CMOS detector actually collects its light sequentially, or can collect the light sequentially. And so if you time the collection of light by this CMOS to the scan, essentially it's like acting like a confocal aperture in a way, or line scan aperture at any rate. So you're as the beam is scanning through the sample, you're collecting photons in this narrow band of active pixels. And when you do that, you end up getting much, much higher resolution. This is the same volume collected by conventional light sheet microscopy. And this is by this actually swept light sheet microscopy. So you can see much, much higher resolution. So this is 330 by 330 microns. And when you do that, now all of a sudden you can collect it's still not an enormous field, but you have 330 micron field at 0.5 by 0.5 by 0.5 micron resolution, which is isotropic, which is also very cool. So it's a very exciting technique. Another issue that has in light sheet microscopy, it has to do with how you present the sample. For most of us, I think, certainly for me, if the idea is you have to project the light from the side, then somehow the sample has to be up on a sample, it can't be sitting on glass, right? And so if you've got an embryo, you can mount that in agarose and rotate it around. If you've got some chunk of tissue imaging with the like a twins fleck, you have to have some mechanism whereby this is maintained up in the column, typically mounting it in agarose. This is the Rube Goldberg thing that the Vision Biotech system says you actually put this tissue in a clamp and get it up into the light column. This is fine, but it's finicky, it's troubling. And the other thing, it's completely incompatible with slide mounted tissues. So you'd like to be able to look at slide mounted tissues. And in fact, this guy that we're working with in Washington, he is all about doing this for pathology. So how can you make light sheet microscopy work for slide mounted samples? There's a lot of different approaches. I'm not going to talk about any of them. In general, all of them, except for one, have concessions to field of view. So these systems in particular are great for small field of view. And we'll talk some more about these systems later. This system right here, the open top light sheet microscope system, is the system I'm going to spend some time talking about. This is a system that John Liu came up with in Washington. And the way that this works, you take everything and put it underneath the slide. You get tissue mounted on a slide. You get an illumination objective from here, and then orthogonal to that, you have your collection objective, and you just translate the sample through the slide, through this cross-sectional area that you're imaging from. And this gives you kind of a sense of what that looks like. This is what you'd be imaging like by conventional techniques. This is what you'd be imaged with the light sheet microscope system. This is how it works. This is a movie that didn't play this morning. Just to give you a sense, for those of you who've done it before, it is astonishingly easy to do this. Some guy with purple hands is going to come in and mount the tissue just by dumping it into this chamber. And then you patrol around and you identify the region you want to image, and you collect images of it. So this is a very, very powerful technique. It's a very fast way of getting through tissues without having finicky needs to how you mount the tissue. And to my knowledge, it's the system that you can use for imaging large slide mounted tissues. Here's an example of a volume that was cleared from a TDE cleared mouse. And we all know what clearing is now. This is from a brain slice using this system. And again, you've got enormous scope, but you also have terrific resolution. Okay, in the interest of time, I'm going to cut this short. This is an interesting application. This is Josh Vaughn at Washington does expansion microscopy. Expansion microscopy is where you isotropically expand a tissue. So if you want to know, another way of approaching super resolution microscopy, well, you can approach super resolution microscopy by either breaking the diffraction limit and imaging finer than the diffraction limit, or alternatively, taking your sample and making it bigger. And that's how expansion microscopy works. Well, that's not how it works, but that's what it does. And the problem with that, of course, is that then you've got this enormous volume that needs to be imaged. If you're doing 5x or 10x, the volume goes up as the cube of those dimensions. And so all of a sudden you're facing an issue of imaging large scale images. So this is some kidney tissue, which was imaged by light sheet microscopy following tissue clearing. And this is one of these groovy movies that Adam Glazer put together.
So Ken, this is
Yeah.
Diane. I'm wondering, at the centers around the country, like does Indiana or Cincinnati or Yale have this capability?
What I don't mean, Yale certainly does. I don't know if it's something that's offered as a service. We are in the process. I think next week we have a grant being reviewed that we hope will get us one. I've got another grant I'm going to be submitting for a shared instrumentation grant, which would get us one like a year later if it were funded. So we have a system on campus. It's strictly for VA. I'm not aware of anybody in the CCEH who has one of these things, but I could easily be wrong. To be honest, a lot of these things are languishing because people buy the system and they don't end up getting used because they're not that trivial to use. So if people are interested in this, I suggest that they explore their institution. I started this movie before I should have. All right, so what I wanted to point out here is that light sheet microscopy is, let me go back, I'm sorry. Light sheet microscopy speeds up the volume of imaging, but you don't have terrific resolution. If you're imaging an entire mouse brain by confocal microscopy at 5 by 5 by 5 micron resolution, it'll take 37 hours. Do it by light sheet microscopy, it'll take an hour. If you want to do it at 0.5 by 0.5 by 3 micron resolution, it'll take 228 days by a confocal microscope, and it'll take 2 weeks by light sheet microscopy. It's a lot better, but 2 weeks still is not a non-starter. So the approach that John Liu came up with was this hybrid system. And that's what you're going to see here. So here's the volume imaged at the lower resolution. And you can see you've got various blood vessels, you've got nuclei. And then after that wand is what happens when you've applied this, you've imaged through this second light path. So that's the low power resolution, and that's the higher power resolution. And this is mediated by something called a hybrid design, and I'll explain that in a second. So in the way that this works, and this is the conventional light path. You've got a light sheet penetrating through here. You're imaging here, very high speed. Down here, you've got another objective, and you're imaging diagonally. Essentially, you're imaging a plane that's not orthogonal to the objective, which means you're going to have to translate this diagonal image into an orthogonal through a second focus. So you're taking this focusing and turning this into an orthogonal plane. You collect an image there. This has much higher NA, lower depth of penetration, and it takes longer to collect. The idea of the strategy is that you take this hybrid strategy, you image the entire volume you're interested in and at low resolution, and then you follow up later and you've identified regions of interest that you want to go at in higher resolution. And this is a nice, it takes a while to get through this. So if anybody has any questions, we can talk about it, but this demonstrates how it works and why it works. So this is the design they came up with, the light sheet microscope. And I don't mean to be selling the system because I'm very gung-ho on it. They designed this thing so that it can image tissues on slides. And at some point, he'll start talking about the hybrid design. All right. So again, this is a volume I showed you a moment ago. This is the image before with one light path versus the other light path. So the idea, and I think this is fair, and the example that they show is a good example. You don't necessarily need to image an entire organ at sub-resolution, subcellular resolution, but you would like the opportunity to inspect certain areas. So here's an example of going in and looking at individual neurons, for example. This is a really cool example because these are mets. These are cancer mets in the brain. And you can use the low power mode to identify the location of those mets. And then you go in and you image those higher resolution to characterize them at the resolution you want. So this hybrid strategy allows you to kind of capitalize on the ability to survey an enormous volume, like the entire brain, find every example of the thing you're interested in, and then collect a high resolution image of that thing with the same instrument. So we're real excited about it. It seems to offer a lot of the things that we want, because pretty much everything we want to do involves imaging tissues on slides. So I see it as a very powerful advance in light sheet microscopy. This movie will go on and on. If you're interested, I can point you to the, actually, I've got references at the end to show you. So this is a list I put together this morning of light sheet microscope systems that are commercially available. There are more. I'm sure there are more, but these are the ones I know of. Best known is the Ultravision 2. That's set up for large scale imaging. I don't think you can really do high speed, high resolution imaging with it. Same thing with the Zeiss Light Sheet 7 for large scale imaging. The Alpenglow system is a system I was just talking about. You can do large scale slide mounted tissues at low and high resolution. The Leica SP8, that's that goofy system that has the two mirrors hanging off the objective. It's pretty much for intermediate scale. We found it really hard to work with. Intelligent imaging innovation makes this Marianas light sheet, which is actually designed for small scale live cell imaging. Bruker makes 5 different designs, each optimized for a different thing. So either tissue imaging, high speed imaging, whatever. Applied Scientific does as well. Lattice light sheet microscopy was something I was scared of for years because it just seemed way too finicky to want to get involved with it. But it is commercially available, either from Intelligent Imaging Innovations or from Zeiss. And I encourage you to ask for a demonstration of these. And then the other thing that I don't think people necessarily know is that light sheet microscopy is something that's just not that hard. It's not that difficult to put together. And in the interest of that, there's this Open SPIM project that describes everything you need to know to build one of these things. All the parts, it has the parts list. It tells you how to assemble it. It tells you how the software, it provides you with the software to do the deconvolution and the image analysis. Jan Huysken, his lab, he's all about democratizing light sheet microscopy. He has this Flamingo website that describes all the things you need to know, but I think provides you with a lot of it. And then finally, this very, very high-tech system from Rito Fialka. It doesn't provide the design, but he has a very, very elaborately described technique and system and again, in the interest of democratizing it. So there's, unlike a lot of things, like confocal microscopy is not for the faint-hearted to develop on their own, build their own system. Light sheet microscopy is not that difficult to put together if you've got, some skills and some time. So those are the availabilities. This, I guess, is going to be recorded. So here's some papers I recommend you look at. Some of them are review articles. Some of them are very specific to the stuff I talked about today. But all of them, I think, are really, it's a very exciting technology. I wish I could sum up by saying how you can come and do it at our center, but unfortunately, I'm not going to know about that for probably another month or so. But we look forward to working with you. So, with that, if anybody has any questions, we've got 5 minutes.
I have a question. So, most of what I've seen about light sheet microscopy is outside the bone marrow. Right, and this is because of the limitations of the tissue. Have you seen any recent papers where you don't have to permeabilize and stain for days to visualize the marrow?
Well, I mean, and I am not in a position to talk about this, but many of the volumes I showed you, all the volumes I showed you, showed the bone marrow. I don't know how the tissue is prepared to access the bone marrow. I don't know if you're imaging through the bone. Yeah, I don't know that. I mean, I'd recommend that you check out some references to find out if anybody has successfully managed to do that. I mean, people clear bone. I don't know if they clear bone to the degree that the calvarium is well imaged. Certainly not imaged at the kind of quality that your work has. And the other issue then again is going to be getting your antibodies or any other reagents into the tissue. So I can't really comment on that.
Okay, thank you.
Sure.
Hi,
Okay.
hi, Don Kreitzer, Yukon Health. We work on implantable medical devices like glucose sensors and
Yeah.
fusion sets and stuff. And one of the things that we're very interested in is many times, like the sensors, the tissue reaction causes loss of function. So there's
Right.
a lot of things trying to understand tissue
Yeah.
reactions and stuff. Are you aware of anybody that's tried this on a implantable device, like a sensor, which is like plastic and it's got coatings, but it's like the thickness of a fat pencil line? Do you think that would be possible to?
So you're talking about, where would you be imaging that from? I mean, so you would be.
Well, you put these in the skin of people with diabetes, and it's supposed to last for a long time, but it usually fails because the tissue reaction, it's a foreign object, just like a bacteria or a splinter in the body attacks it, but that attack causes it to fail, biofoul. Do you think this could be used to visualize the tissue reaction occurring around this, basically what's like a little splinter that's good for you as opposed to a splinter not so good?
You know, I don't. You're talking about imaging in a live animal, correct? Or you just want to see? You'd be happy just collecting tissue and then imaging the reaction to the tissue.
Right at this point, to be able to collect the tissue and put it on a slide and be able to image it in 3D, just to kind of say, even if it was sequential, like day, 2 days, 5 day, kind of thing, to see the reactions by using probably either immunofluorescence or even, mutant mice, knock-ins, knockouts, that kind of stuff to start with.
It's interesting you bring that up. I mean, the thing, there's no reason you couldn't do that. But the thing is, at that point, what you really want to know is the advantage of light sheet microscopy is that it's going to give you scale, right? You're going to be able to see the entire object, perhaps, and see interactions with it. But and this is a little self-advertisement here, if I can get this to display. So I'm not sure why you'd want to use light sheet for this necessarily.
Because these are big objects, and histopathology, that stuff doesn't.
How big are we talking about?
Well, like I say, they are probably, gosh, I don't even know what the size. Like I say, they're maybe an inch long and maybe an eighth of an inch wide, and it's embedded in the skin of these patients. Or another one we're working on are stents for cardiovascular, to look at those reactions. And the problem with them, they're metal. It's hard to section.
These would be animal tissues, right?
Well, we get them from people.
Okay.
In the sense of sensors with punch biopsies that take the whole piece. Or the first, so in principle, there's no reason you couldn't image these things, although the solid object in the light field is going to be a problem. I thought maybe you rotate it and sort of like going around a spindle.
You probably could do that. Man. So if you had
I don't want to take any more time. Thank you. I'll follow up.
Well, actually, I want to share one image with you. And that is, we've been thinking about the foreign body response inadvertently. I didn't want to study the foreign body response, but I do intravital microscopy, right? And let me see how I share my screen again.
Again, this is the one. Can you hold sharing your image? We have one more question.
Okay, sure.
Then we
maybe. So Don, is that your name? I'm sorry. Good. How about you hang around and everybody else can leave, and I can show you the stuff that I've got. All right, go ahead.
Thank you, Diane. Thank you, Ken. Some of the messages you delivered today is really something I needed to hear, especially for the one you were saying it's not that hard. I needed to hear that. Why am I saying that is, from my own research, we're interested in the cellular dynamics. So for example, the rapid dynamics happening on minutes level, for example, the ERK dynamics or NF-kappa B dynamics, this kind of transcription factor in and out of the nucleus, looks to me or feels to me that to image such live transcription factor dynamics, the light sheet microscopy would be the way to go, because otherwise,
if there's any way. I mean, I think that I don't know what kind of signal levels you'd have. So whenever you're imaging something like this, it's a question of how fluorescent the thing is and how fast you need to characterize it. And so if you've got something that's not very bright and it's moving around really fast, nothing is going to work. But I don't think there's going to be anything, and I didn't emphasize the cellular level stuff at all. And so pretty much I really emphasized tissue. But the point is,
why I wanted to ask.
yeah. And so a lot of the systems are really oriented about high-speed cellular, like 100 micron area regions. And so I think if there's a way to image it, I would think that the lattice light sheet would probably be the system. But again, if anybody can. So, this might be one of those things that you invite Zeiss in to demonstrate their system. And if you've got fluorescent transcription factors, see what you see. And then you've got the basis of shared interpretation grant.
Yeah, those images or those dynamics have been imaged very successfully in the cultured cells, but never for example, in marrow.
Okay, so you see now you're talking about imaging doing intravital microscopy then.
Actually, I was thinking whether it's possible to take the marrow core out, just to image that core. Live.
Yeah, I have no idea how happy the marrow is outside the body. I mean, that would be the issue there.
I can tell you that it's extremely unhappy.
It's what?
Unhappy.
It's very unhappy.
Unhappy, yeah. Well, there's unhappy and there's unhappy. It's like a lot. I can tell you, we do a lot of intravital microscopy. The mouse isn't pleased, but it's the question of the biological question you're looking for. Yeah. And so, you're going to if you do that, you're going to have to somehow validate the system you're looking at. It's not hopelessly deranged by isolation.
Sure. So we do have on Yale campus, we do have the Bruker system. But it's scary to all the hematopoietic people. But I think as our imaging core will start bringing them in.
Yeah, particularly if Jorg helps you with that. I mean, and I think when you say you have the Bruker system, like I said, I think there's 5 different systems. So you might have the wrong one. Who knows?
I'll have to ask the right people, because the Jorg himself is not directly responsible for running the light sheet. So I'll have to figure that out. Okay.
I don't know. I'm knocked out by what they can do with the light sheet. The lattice light sheet system, it just has such capabilities. There's so many reasons it's a good idea. So,
well, thank you.
Sure. Well, thank you for attending and asking a question.
I'm going to stop the recording now, and then Dawn and Ken, if you want to continue to talk, you can.